So over to Catherine.
Thank you. So I'm going to try to follow up on the difficult act after the beautiful talk from Bianca and this beautiful music. I think one link might be this extraordinary sculpture from Camille Claudel, which I think reflect very well love and some of the themes we heard. And what I would like to discuss today is some of our data that illustrate not only love, but not to love. How is that choice being made?
Move a bit to the center, that's all. Just, yeah.
Okay. Both from between adults, but also on this other beautiful sculpture from Camille Claudel, between adults and an infant, following some of the theme that Bianca beautifully developed. So in my lab, we're interested in understanding how social behavior is generated. And we're interested to understand this in a very brain-wide manner, which is that we want to understand what are the signals that are detected in periphery, how our sensory system detect particular cues, and how these cues then processed within the brain by sensory areas, by central areas, according to the state of the animal. We heard virgin males versus mothers, but also males and females, in order to create behavior. Now, there's something quite interesting in the difference between the sensory processing and the central processing, which is that the detection, the cues that are detected that lead to particular social behaviors are typically extremely idiosyncratic. They're extremely species-specific. So, for example, this bird here recognizes another male of the species just by this little black spot here. We'll see how mice use particular cues, pheromones, and then humans have another set of cues that they obey or rules that they obey in order to engage in social interaction. Now, it makes a lot of sense because for an animal of a given species, it's really critical that they interact with another animal of their own species. So therefore, the cues, the signals that are detected and interpreted are extraordinarily species-specific. However, the central circuitry within vertebrates or within mammals, within mammalian species, are thought to be highly conserved. So, for example, the circuitry that drives maternal behavior, here seen on a bird, on a mouse, on a human, are likely to have very similar architecture. So the detail is probably going to be a little bit different, but overall, the rules that scientists are able to uncover, such as what we've heard from Bianca, are likely to be highly conserved from a bird to a mouse to human. So in my talk, I will first discuss this peripheral aspect of sensory detection on the particular phenomenon of pheromone detection and the control of adult social behavior in mice. And in the second part, I will discuss our efforts to understand the circuit of parental care, but also adult reject of infants. So these two opposite behaviors towards infant, love versus reject. So let me talk about first the detection of social cues in mammals. What you see here is called the Flehmen response. It's the animal are retracting their lips and inspiring, trying to detect social scent from animal of their own species. And why do they retract their lip? Well, because this particular grim that is being observed enables social cues to enter these particular olfactory neurons, olfactory organ called the voronasal organ. Now, the VNO does not exist in higher primates, does not exist in human. I'll discuss this a little bit later. But in most mammals, in most terrestrial vertebrates, actually, this voronasal organ operates and detects these social cues called pheromones. Now, what is very interesting about the voronasal system is that these neurons that are located in the cartilage in the middle of the nose, in the the septum, these neurons are connected to a particular circuitry in the brain, to an area of the olfactory bulb called the accessory olfactory bulb, the medial amygdala, and then centers in the hypothalamus, a deep brain area that is involved in the control of aggressive behavior, mating behavior, parental behavior, all sort of social behaviors. Now, what is fascinating, or what fascinated me when I start to work on the voronasal system, is as you can see, the processing of sensory information entirely bypasses cortical areas, cognitive areas. And so the idea is that these social cues are detected and trigger instinctive social behaviors such as sexual behavior or aggressive behavior. And the system differ and contrast from the main olfactory system, in which the main olfactory epithelium that is located in the posterior recess of the nasal cavity send information to the main olfactory bulb and then to a series of areas that are globally called the olfactory cortex. So as you can see, an area that drives a system that drives instinctive behavior versus a system that drives cognitive responses to smell. Now, interestingly, these two systems use very different molecular machineries to detect sensory cues and convert them into electrical signals. In the voronasal system, neurons express a particular ion channel called TRPC2 that is only expressed by these neurons in the entire body, whereas the neurons in the olfactory epithelium express a different ion channel, which is an olfactory gated calcium channel. Moreover, the receptors that detect sensory cues are also distinct. The voronasal organ express this family of receptors called V1R and V2Rs, whereas in the olfactory system, Linda Buck and Richard Axel discovered this large family of olfactory receptors. So we were very interested in trying to understand how the voronasal system in mice trigger social behaviors. And the expectation was that, well, you know, what the VNO does is detect these love signals that are floating in the air, detected by the VNO, and then triggering sexual behavior, mating behavior between mice. And what we found was very, very different. And I'm going to tell you that story in the next few slides and then lead you to our more recent work. So the idea is to use the power of mouse genetics to ablate genetically the function of the voronasal organ and observe the social behavior of animals. We started with experiments done in males and then experiments done in females. And each of these studies tell us something very particular and very interesting about how the brain copes with social behaviors. So in males, we first abolished genetically the expression of the TRPC2 ion channel. So in these animals, males do no longer have a functional voronasal organ. They are, if you wish, pheromone blind. So what would happen to these animals? Well, our expectation was that this animal would not mate. But not at all. Male and females were able to mate perfectly normally. But when we tested then another social behavior, aggressive behavior, by putting two males in the same cage, then something strange happened, which is that the males actually mated with each other. Oh my God, what did we do? And so we decided, we say, well, maybe these mutants are hypersexual. So let's put in a cage both a male and a female intruders together with a mutant and see what's happening. And what's happening is that the mutant male actually mate with the female, and then with the male, and then with the female, and then with the male, with equal frequency, suggesting something quite interesting, that these animals are actually not able to tell apart a male from a female. And so there are two really interesting phenomena here. One is these animals are able to mate, meaning that they use other sensory signals to tell them to mate. what could these signals be? Well, probably olfactory. That's the next possible target. So we generate a mutant that does not have a functional main olfactory system, and this time there's no mating behavior whatsoever, neither with males, neither with females. So what did we learn here? Well, we learned that first, both the voronasal organ and the olfactory epithelium work together to control mating behavior. So it is not true that the voronasal organ is the only organ detecting pheromones. The main olfactory system is also able to detect pheromones, and together they control mating behavior. And the role of the voronasal organ is very particular. It is to discriminate between males and females. So a very particular role in the control of mating behavior in general. So that was for males. And you know, when I started my lab as a woman investigator, I said, I'm going to do half of the experiments on male behavior and half of the experiments on female behavior. But it took us more than 5 years before starting this experiment in females, because the literature is extraordinarily poor in describing female behavior. Well, basically, there was no interest in female behavior. Male behavior always seems to be so much more interesting. Anyway, I had trouble to identify or have a lab member interested in female behavior, and he came my former postdoc, Tali Kimchi, who is now a professor at the Weizmann Institute. Tali was trained to observe wild animals, actually blind mole rats at the University of Tel Aviv, and she said, you know, even if the behavior of the female mutant is subtle, I'm trained to observe animal behavior, and I'll see it. And in fact, what she saw was not something very subtle, as you will see, which is that we looked again at the behavior of the TRPC2 mutant female, and also to make sure that our observation were not due to developmental effect, we also removed the voronasal organ surgically from an adult female. And what did we see? Again, something extraordinarily surprising, which is this female now behaved like a male. Not only it attempted to copulate with a male, but also with females, exactly as a male typical copulatory behavior, suggesting that there were two things happening. One is an absence of sex discrimination. Here again, the behavior is the same towards males and females, so this is something we've identified with males. But in addition to this, it literally is the sex identity of the female that has been changed. And so what we learn from this is that the voronasal system gates sex-specific behaviors. It tells a female to behave as a female, and if the voronasal system is no longer functional, now some other behavior circuits are uncovered. And in that case, in female, male behavior circuit. There is a conclusion from this, however, which is that our experiment demonstrates that some male typical behavior circuitry exists in the female brain, right? And so the next question, therefore, was, well, if we find male typical circuitry in the female brain, are there female typical circuitry in the male brain? And so this is what we decided to address next. But let me first, as I mentioned, sensory detection is very different across animals. And so as we were discovering, making these very surprising findings, I got really, really worried that people would extrapolate to human and ask me about, well, what does that mean for human sexuality, basically? And I, you know, at the time I was a junior faculty, and I was terrified that journalists would just take this and make a big story about the genetic root of homosexuality or something, which I absolutely did not want to go into. And so I started to think about, how am I going to respond? And I thought, well, maybe what I should do is to look at other animal species. How do animals recognize the sex of other animals in general, and try to get some inspiration out of this? And I found some really fun examples. So this is a parakeet. And one interesting difference between males and females is this blue dot on the top of the beak of a male. Now, if you paint this blue dot in white, so it disappears. Now the other males are going to try to mate with this male without a blue dot because they think it's a female. Similarly, if you paint a blue dot on the beak of a female, the male are going to attack that female with a blue dot because they think it's a male. And so this is an interesting sign of what is a male and a female. Similarly, this American flicker, this is a female and this is a male, and animals within that species are able to tell apart a male from a female thanks to this little black patch of feather on the face of this male. So again, if you mask that patch, then the other males will try to mate with that animal because they think it's a female. If you paint a black patch on the face of that female, the males think it's a male and will attack it. And so Timbergun, who is this fabulous ethologist, called these the badges of masculinity. So I have a mustache, I'm a male. I don't have a mustache, I'm a female. And so I think that what we found is that the pheromones are the equivalent of these badges of masculinity. They are the olfactory badges of masculinity. And so now, obviously, you wonder, well, what about us? Well, let me tell you about us. First of all, the TRP ion channel, the TRPC2 ion channel that I described to you is a pseudogene in human, meaning it's a non-functional gene. What you see, these little yellow triangles here, are deleterious mutations. Only one of them would make that gene non-functional in human. 9 of them make that gene dead. In fact, it's, yeah. So, and the interesting question is, when, during evolution, did that ion channel, in other words, the function of the voronasal organ, disappeared in human? And as you can see, the split is here between the old world monkeys containing the apes the apes, human are here, the apes and the old world monkeys, and the new world monkeys. And this is interesting because that evolutionary split also corresponds to the duplication of the red and green opsin genes. And that means that at that split here, the animals that are located in the bottom of that tree have enhanced visual ability because they are able to tell apart red from green, whereas the animals that are above are dichromat, so they're unable to tell apart various colors. And so what that means is basically the animal here, the apes and the old world monkeys, rely enormously on visual cues, auditory cues, odor cues, and olfaction became way less important. So what I told you about the voronasal organ is pertain to the animal here that are dichromat. And so, in other words, there's no functional voronasal system in human and higher primate. There's higher reliance on sensory cognitive cues as well as internal representation. But I don't think that completely makes human escape from instinctive representation, instinctive behavior. And I would like to point to one phenomenon that we all know about, and that, in my view, triggers to the fact that sexual arousal can be triggered by sensory cues in human. It's the phenomenon of pornography. There are millions and millions of dollars that are being made by creating these videos that show through visual and auditory cues trigger sexual arousal in human. So again, just for your thinking, I don't think that we all rely on cognitive and rational thoughts. We also are a recipient somehow of cues that release behavior. So let me now switch to parental behavior. As I mentioned, we were very intrigued to see this male typical circuitry in the female brain, and that really to some extent prompted us to look whether there could also be female typical behavior in the male brain. And so what type of female typical behavior were we going to look at? Obviously, maternal behavior, this beautiful behavior displayed typically by female mammals. In mammals, it makes a lot of sense for the female to be the primary caregiver. Through gestation and lactation, female invest enormous resources for the well-being and the survival of their offspring. So obviously, they will make sure that these offspring can survive. The contribution of males, however, is quite variable, and in a significant number of mammalian species, males are aggressive. They attack infants and kill them in a phenomenon called infanticide behavior, which in animals is not a pathological behavior. It is thought to be an evolutionary conserved behavior that by which foreign males or virgin males will kill the offspring of other males, which will provide them an advantage by both eliminating the offspring of rivals, as well as having females now re-enter the sexual cycle, and therefore they can sire their own offspring. And so in mice in particular, females are parental, whereas a virgin male virgin males are infanticidal. So as Bianca mentioned, mothers are these excellent, display this very quick maternal behavior. Virgin females are by far not as good, but if you leave them enough time, after a while they will retrieve the pup, get trained, and be a caregiver as well. In contrast, virgin males are very systematically infanticidal. So we know how important parenting is in human. It's essential for the emotional, physical well-being of individuals. Humans take years and years, maybe lifelong care and sacrifice. And in human, there's this really interesting structure of parenting in which not only the parents, the extended family, the friends, even the social structure are involved for the well-being of infant. But in a significant percent of mother and even fathers, and frankly, these numbers are staggering. When I discovered these numbers, I was appalled. I do not know of any mental illness that is that prevalent and for which so little is done. Women's diseases tend to be underappreciated, but these numbers are really, really frightening. Now, the symptoms of postpartum depression are extreme worry from the mother's side, dread, feeling of failure, and difficulty in bonding with the child, with frequent thoughts of harming a baby that are not pursued by action, but that are debilitating just because of these thoughts of harming a baby. And so here is a testimony of a woman with postpartum depression. I have broken down, I don't know how many times, or I'll get angry about the crying, then sad about being angry, and then guilty, like, why do I feel so sad when I have a beautiful baby? The emotions are insane. These could be said by most women with postpartum depression, but the reason I'm providing this particular quote is because of the woman who pronounced this word, Serena Williams, who epitomizes woman's strength. And so this is just to say that postpartum depression is not a weakness, it's an illness that needs to be taken care of, and it's just really important to address this issue. Now, in extremely rare cases called postpartum psychosis, which affect about 1 in 1,000 new mothers, then the symptoms and this obsessive thought of harming a baby can be followed up by action, and therefore these require hospitalization. But this is an example of how important the understanding of care versus the non-care of infant is important. How is it encoded in the brain? What are the neurons involved? What are the circuitry involved? And how is circuitry regulated by various internal as well as environmental factors? So let me first talk briefly about the sensory control of infant care. I told you we're interested both in the signals and how the signals are processed internally. We heard beautifully from Bianca the importance for parenting of auditory cues. And in some circumstances, olfactory cues are also important in close proximity. And as I mentioned, mothers are extremely good at taking care of infant, even not their own infant. And virgin females, when put in presence of infant for a sufficient long time, are also caring for infants. So females are just instinctively parental when put in presence with infant. And again, they are parenting not only with their own pups, but the pups of others with these communal nests that are established in the wild. Virgin males, by contrast, as I mentioned, are reliably infanticidal. And so that led us to wonder what is driving infant-mediated aggression in males, and could it be the voronasal organ? This organ that we saw is able to gate the sex specificity of behavior. So we decided to look at mutant males, at virgin males that are mutant for the TRPC2 ion channel, and indeed, these VNO mutant males no longer attack infants. In fact, they are extraordinarily maternal or paternal. And so that really confirmed our idea that the voronasal system gates sex-specific behavior and that the male brain might also have maternal circuit. Now, why would male have maternal circuitry if they are infanticidal? And so here, by looking at the old literature, we found something quite extraordinary, which is in fact in mice, males switch from infanticidal to parental behavior after they mate with a female. And that switch does not occur right away. It occurs after 3 weeks, which corresponds exactly to the gestation time in mice. In other words, a male mates, any pup that comes in the next 3 weeks are unlikely to be his own, so he will attack it. But after 3 weeks, any pup around could be his own, and therefore he's caring of them. And let me show you, this male was an infanticidal male, has mated with a female 3 weeks earlier, and as you can see, has built a nest, and now is retrieving these pups that are not even his own pups. These are 5 pups that we put there from another male, and as you can see, you would not be able to distinguish that behavior from that of a female. And as you see, such a good father is making sure nobody is left behind. And when he's satisfied, he will go back to the pups, crouch around them, just an extremely good father. So what that tells us is that there is this really fascinating plasticity in the male brain. And that is quite interesting also because from an evolutionary standpoint, if you look at insect and fish and frogs and all sorts of species in which only the females take care of infant, only the male take care of infant, or both of them, some of them are genetically very similar. For example, my friend Lauren O'Connell, who is a faculty at Stanford, looks at these poison frogs. This is a male here in blue, and you have a little tadpole here that the male is carrying and protecting. This is, a biparental species in which also you have a tadpole, and that's also a male. And in this species, only the female take care of the tadpoles. And so it's very likely that in these 3 species that have different display of parenting, the circuitry are actually there, but gated differently. So from an evolutionary standpoint, the idea that parental circuit exists both in the male and the female brain, although they might be displayed differently according to the genetic background, is, I think, a really interesting idea. So if the voronasal organ triggers pup-mediated reject and attack, what are those cues? Can we identify them? Can we learn something from there? And this is what I'll describe now is the work of Yui Sogai when he was a postdoc in my lab, now a PI at the Sainsbury Center working on neural circuitry. And so Yo decided to look at male attacks. And what he realized is that male will attack a dead pup, suggesting that movement or temperature were not important. So he decided to make silicone dummies. And these silicone dummies, when they were coated with pup urine or pup salivary extract, elicit exactly the same marks of attack as a normal pup. So in this system, he's able to recapitulate the trigger of attack. And he wondered, what is the animal recognizing that triggers attack? And he decided to look at the morphological features because, as I mentioned, movement doesn't seem to be involved, temperature either, the animal attack in full darkness, so maybe they touch the pup and are able to recognize that this is a pup together with the pup sense. And so again, with the silicone dummy and salivary extract, he showed an attack when he had what he called a brick, which is the same size but completely the wrong shape. There was no attack. When he made what he called a blob, which has the shape of the head, the curvature of the body, and he coated these with salivary extract, there was also no attack. However, in the structure that he called hybrid, which is basically the brick with the legs and the tail, then there was attack. So somehow the male is able to recognize, and you can see because with their whiskers, they touch the legs, they touch the tail, so they are able to recognize the specific features that tell them this is a pup and I need to attack. But they do that only in the presence of salivary extract. And so Yeo is a biochemist extraordinary, and he was able to actually identify both the receptors that detect cues that lead to attack, as well as the particular chemicals that trigger the attack. And again, we learned something very interesting here because the two chemicals that he was able to identify, this submandibular gland protein, which is salivary gland protein, as well as a component of blood, are components that are both displayed by the pups as well as the mother. So the mother licks the pup and coat the pup with this salivary protein, and during delivery, during birth, generates some blood that sticks around the cage. And both of these cues are used by the male to recognize he is a mother and here are pups and I need to attack. So I think something really interesting. So let's go back now to the circuitry of parenting versus infant-mediated aggression. We ask, what are the neurons in the brain that triggers parenting and that triggers infant-mediated aggression? So again, as I mentioned, fathers are parenting, virgin female are not very good at parenting, but they do it after a while, and then mothers are superb parenting, and then males, virgin males are infanticidal. Now, I should say females are also occasionally infanticidal. Virgin females are sometimes infanticidal, but also in case of stress, so when there's too much noise in the mouse facility, for example, or in the wild, females that lack food or that are in the presence of predators very often decide to kill their infant and wait for better circumstances to be more successful mothers. So Herbert Wu, a graduate student in the lab, now has his own lab in New York, decided to look at neurons in the brain that are specifically activated during parenting. And for these, he used a particular readout, which is the expression of a transcription factor called CFOS that is elevated when neurons are firing. So the idea is you have an animal, a male or a female, interacting with pups, then you sacrifice the animal after half an hour, and you look for brain areas that are CFOS positive versus CFOS negative in non-parental animal. And what he found is that there is this particular area of the brain called the medial preoptic area. This is an area deep in the brain, in the hypothalamus. And in this area, you can see that parental males and female all have very strong CFOS expression, whereas infanticidal male do not. Now, there's nothing surprising here. The medial preoptic area has been involved in maternal behavior for decades by a lot of very beautiful work in rats and in mice. But what we're interested in is what is the identity of these green neurons here? They are surrounded by negative neurons. What are the CFOS positive neurons? And what Herbert found is that the neuropeptide galanin was expressed by a substantial number of CFOS positive cells. So this is not the perfect marker. Some galanin cells are not CFOS positive cells, some CFOS positive cells are not galanin positive, but this is the best we could have at that time. And I'll show you in the last few slides that we've done way better since then. So the idea is that, well, if these galanin cells are activated during parenting, maybe they are involved in the control of parenting. And so we performed two key experiments. One is using transgenic tools to kill specifically galanin-positive cells in the preoptic area. Now these are animals that no longer have these galanin cells, and we tested the maternal behavior and the paternal behavior of these animals. And we realized that killing these cells abolished both maternal and paternal behavior. So these neurons seem to be essential for the display of parental behavior, both in males and in females. Now, what about a gain of function? Let's try to take an infanticidal male and specifically activate these neurons. What we would get? Well, what we get now is that these formerly infanticidal males display paternal behavior. In other words, these neurons seem to be a critical node for the control of parenting, both in males and in females. So how do they work? Joe Nickell, now an investigator here at the Crick Institute, decided to look at the input to these neurons, at the output at these neurons, and then how different phases of parenting behavior are controlled. And what he found is, first of all, these neurons that you see here in the center receive a lot of information from a lot of brain area, around 20 brain area. So, you know, the function of these neurons is complicated. It receives a lot of information, and they send a lot of input to other brain areas as well. And what he found is that the projection of a particular MPOA galanin neurons is precise. It goes to only one particular brain areas. In other words, these MPOA galanin neurons are subdivided into modules that presumably each deal with a subset of what parenting is about. And this is what he tested directly. He found that MPOA galanin neurons project to the paraventricular nucleus, the area that Bianca described as containing oxytocinergic neurons, and indeed, they send projection to neurons expressing oxytocin. In other words, they drive the release of oxytocin as well as other social peptides. Journey also decided to stimulate a particular subset of projection to an area called the pericaudal gray, which is an area involved in motor control. He found that by stimulating the PAG projection of both males and female together with pups, he increased grooming, increased the motor execution of parenting. Then we decided to look at other projection, and one area that was of very high interest to us is the VTA, or ventral tegmental area, because this is the area that secretes dopamine, and dopamine is thought to be essential for the motivation of animals to engage in behavior. So what would happen if we specifically activate VTA projection? Well, we found that there's no increase in parenting. So we had to think a little bit differently, and we thought, well, you know, female go to make huge effort to reach their pup. This is a mother. Her pups are in this tall beaker here. And when the female realizes that, she really wants to reach them. She cannot through the glass. And she's then going to realize that, oops, you know, maybe she could reach them from the top. And as you'll see, she's going to go through some pretty tough acrobatics in order to reach them. And one by one, she will indeed be able to catch them. And so that made us think that, well, maybe what we should test is the extent to which the female or males will go through to reach their pups. And so we designed a little assay in which we had a barrier and see whether stimulating the VTA projection would enhance the cross of the barrier by males and females. And indeed, it does, suggesting that this particular projection indeed increase parental motivation. But there's something that we observed also that was extraordinarily interesting, which is that if we do this experiment, if we stimulate VTA projection in mothers and virgin females and in fathers, the males and the female cross the barrier and then are parenting. But if we do the experiment in virgin males, the male also crosses the barrier with a higher probability. But when it reaches the pups, it attacks them. So remember, different neurons project to different areas, and there are a set of neurons projecting to the PAG, and those are the ones that modulate the parental behavior itself. These areas just modulate the reaching the pup. And the second step has to be modulated by the periapical gray. So virgin males do not have galanin neurons driving the PAG, and therefore these males stay infanticidal, although you can trigger their motivation to reach the pups. And then finally, we also modulate the function of another brain area, the medial amygdala, which is involved in adult social behavior. And we found that when we stimulate this projection, now we reduce adult social interaction. So the idea is when you have infant, you no longer go to the pub with your buddies, you stay at home, watch TV, eat popcorns, and so that's the decision you make. You know, you reduce adult social interaction and just stay with your infant. So here's what we found. These neurons that have this particular mode of projection in which each projection is involved in a subset of the parental behavior. And interestingly, Lauren O'Connell in Poison Frog and Andy Bass in Spawning Midshipmanship Fish, so this is a fish, male taking care of his eggs, found that galanin, the peptide galanin or the galanin neurons play a role in the parental behavior in this species, suggesting that maybe more generally these circuitry are involved in vertebrate for the control of parenting. So what about aggression? Well, we looked at the parallel system for driving aggressive behavior. And we found this particular brain area, these neurons in this area called the perifornical area, that are only activated in infanticidal males. Now, these neurons express a particular neuropeptide called urocortin 3, and interestingly, these neurons receive input from the stress area, the voronasal area, and interestingly, they receive input from MPOA galanin neurons, which I forgot to mention are inhibitory. So the idea is that when animals are parenting, they will inhibit neurons that control infanticidal behavior. Interestingly, these neurons are only activated during infant-mediated aggression, no other forms of aggression. And in experiments that I won't have time to detail, we find that when we activate precise brain projection of these neurons, we reduce parenting, we increase aversion, and then we increase aggression towards infants. So in a similar manner as we've done with galanin neurons, we're able to dissect various components of aggression towards infants. So these are the two circuitry, galanin neurons and these neurons that express the neuropeptide urocortin 3, and to some extent they are playing the yin and yang of parenting versus neglect in the mouse brain. Now, we, as I mentioned, our molecular identification is not the best, was not the best. Only a subset of galanin neurons were activated during parenting, and some other non-galanin neurons were activated during parenting. And so we got interested in identifying all the cell types in this brain area called the preoptic area. And we found 70 neuronal population, mostly never described before. And we use an in situ technique called MERFISH that enabled to visualize in situ various particular cell types, so an extremely powerful technology. And because we also visualize this marker CFOS, we can see which particular cell type is involved in mating, aggression, parenting behavior. And what we found is that these galanin neurons that I mentioned, well, in fact, there are many subtypes of galanin neurons, and only one of them, the one in green here, cluster that we call I14, are activated during parenting. And it can be recognized because it's the expression, it co-expresses both galanin and calcitonin receptors, so two markers that give us extreme precision in identifying cell type involved in parenting. We also have this particular cell type that is involved in infanticide behavior. We also identify many other cell types involved in parenting behavior, some that are activated in virgin females, mothers and fathers, some expressed only in mothers, some activate in mother and fathers, and some only in fathers. And one neuron type in particular that we found very interesting is this E1 cell type that is only activated in mothers and is connected to I14. So we think boosts the activity of I14 in mothers and not in other cell types. And so we are excited by this data because we think now we know precisely the cell types involved, and we can explore causal relationship, both molecular and biophysical, in parenting behavior. And indeed, this is what we're doing now. We're imaging mice that are interacting with pups. This is a mouse that has a little microscope implanted. We just put a pup here, and as you can see, now the activity of the preoptic area in this mouse is just becoming extremely strong. And so we can visualize these neurons, and now we can ask, what are the different neuronal populations involved in various parts of parenting? So I'm going to finish there and just summarize by saying we've identified these particular cell types and circuitry that are involved in parenting versus infant neglect and aggression. And we hope to be able to follow these studies by very mechanistic studies of what is changing in these neurons in animals that are parenting, animals that are infanticidal, animals exposed to stress and other environmental factors. And on this, I cited the people who did the work and my funding sources. Thank you.
Catherine, that was spectacular. I mean, when we call something behavior, we always think of it as some sort of parenting. And then Catherine beautifully showed it's composed of little constituent parts, almost Lego blocks. And those can be mapped onto genetically specified circuits in the brain. So going from genes to behavior of various complexity. And that is beautiful. I mean, that's exactly what science should be doing. So thank you, Catherine. We have time for a couple of questions.
Hi. Why was it relevant in the study by Kimchi on female to surgically remove the vomeronasal organ, but not in the previous studies on males?
Yeah, so when we looked at these female mutants and we found that they had this male typical behavior, we were stunned. And we tried to think about various ways this could happen. And one way is the fact that it's well known that when sensory systems are not used during development, they don't develop properly. So one interpretation of this data is simply that the circuitry is miswired in females because they never had vomeronasal input. So we want a female that had a completely normal sensory development, and then we remove the vomeronasal organ. So we know that whatever behavior we observe is due to the lack of VNO and not improper development due to the lack of VNO during development. And so the reason we didn't do this in males is because it hadn't occurred to us at the time. That's the reason. But we did it, actually, and we observed the same thing in males. But we did this later.
Catherine, over the back.
Yeah.
That's loud, isn't it? Right. Thank you for your talk. And a slightly simple and unscientific question from me. You spoke earlier about the sense of smell in animals versus in humans. And there's a lot of talk about people being very interested in like pheromones, and there's like pheromone dating events where people turn up and smell each other's t-shirts.
Yeah.
Is there any strength in that, or as we say in England, is that a load of old bollocks?
So let me first describe a fact that I think is extremely important. About 1/3 of the brain of a mouse is dedicated to process olfactory cues. About 1/3 of the brain of humans and apes is dedicated to processing visual cues. So olfaction is involved to some extent in behavior in human and apes, but it's not extremely important. And so every species has their own sensory modalities. So the T-shirt experiment says that, yes, you know, there are some sort of genetic cues or kinship cues that can be detected by the olfactory system. It's possible. That doesn't mean it's important. Yeah.
Hi, thanks for a beautiful talk. I was just interested, since you talked about these experiments where you're manipulating projections to VTA and to amygdala, and how maybe there's a trade-off between maternal behaviors overriding other rewarding behaviors. So have you or others done any experiments where maybe parents have to choose between maternal actions and other things that might actually give a dopamine spike or something like that?
This is an extremely good question, and these are projects that we're starting now. I'm going to rephrase it even more globally, which is that this area in which the Galani neurons are is an area that is involved more generally into instinctive and homeostatic drive. This is where sleep is controlled, feeding, thirst, mating, parenting, all these functions are controlled there. And the neurons are completely intermingled with each other. And so why are they intermingled? Well, one reason is maybe because the animal has to make decisions to establish priority, and therefore neurons can inhibit each other's function and have their priority established by a dopaminergic signal. And so we are trying to understand how is this hierarchy of behavior and needs occurring. But I have nothing to report for the moment.
We have time for one last question over there.
Hello, hello. Thank you so much for coming on and everything that you do, and it was truly fascinating, so thank you for your work. I have a question. What was really interesting is when you mentioned that even maternal mice, they can decide to kill their babies if they live in a stressful environment, which, you know, they just essentially give up on parenting. My question is there any research done where it was tried to modify their brain or neurocircuits, or stress always takes priority, if that makes sense?
So my former postdoc, Anita Autry, who identified these neurons that drive infant neglect and aggression, is working on exactly that topic. These neurons receive a lot of input from areas involved in stress, and we think that what's happening is that when stress becomes sufficiently high, then these neurons somehow are able to override the parental circuitry. But again, everything depends on the circumstances. You know, maybe the accumulation of different stressors, maybe early experience of stress as an infant will have the mother being more sensitive to stress later. All of this has to be taken into consideration.